Okay, great. Okay, so thanks everybody for coming. Thanks everybody for those of you who are online. So it's a pleasure today to introduce and to welcome James Stafford to give this version of the chair's lecture. We do have one more before we end for the summer, and that's on April 13th, I believe it is. And so Zach Hall will be giving that final chair's lecture. So please stay tuned for that. Add that to your calendars. So I think that'll be an exciting talk as well. So looking forward very much to today's talk, to hear and see the work that James has been doing. Just a few words on James. So James did his BSc here at the U of A in medical and laboratory sciences, and then stayed to do a PhD in Mike Bolosiewicz's lab. The PhD was on antimicrobial mechanisms of fish macrophages, for which he won a Cameron Outstanding PhD Thesis Award. So well done. James then went on and did a postdoctoral fellow, which was NSERC funded at the University of Mississippi Medical Center. And then he came back and joined us as an assistant professor in 2006. In 2012, he was promoted to associate and is now a full professor here in the department. James's work, James has been and is currently the associate chair of grad studies, and we all know that soon there'll be some changes there. But for now, I'm very thankful to have James in that position. He has two main areas of focus for his research. The NSERC-funded research is focused on understanding fundamental mechanisms controlling innate cellular defense responses in vertebrates, and he uses fish as a comparative model for the studies. Secondly, he also is funded from Alberta Innovates and an NSERC CRD, and with this collaborative study, the work is environmental immunotoxicology, and he has collaborators with environmental engineers and in industry. This work is looking at the development of immune cell-based bioassays for the detection, monitoring, and identification of contaminants in water. It focuses on oil sands, processed affected waters, and as well investigates the effectiveness of passive remediation approaches of tailing ponds by a long-term monitoring of demonstration pit lakes. So with that, I'll stop there and welcome James up to tell us about what that he's doing. So,
thanks, Stefan.
You're going to set up.
Yep. Okay, great. Welcome everybody. Afternoon, in person and online. I'm happy to be given this opportunity to talk at the chair's lecture. I hung on as long as I possibly could, and then finally got tagged for it. So here I am. I thought what I'd do today is give you sort of a general overview of the background field, sort of set some context for the type of area of research that we're interested in, and then give you sort of an example of the type of research that we do that allows our students to develop systems to answer or ask questions about immunoregulation. And the focus here is using the fish model, but I'll broaden the intro just to talk about innate immunity in general, just again to set the context. Some of the images that you see here, you'll see again, and they are sort of capturing a cellular process known as phagocytosis, which involves the receptor-mediated binding, capture, and eventual internalization and destruction of extracellular material. And namely, the process is usually used for eliminating microbes from the body. I don't know why I have this picture, but it reminds me now is that this looks very much like the phagocytic process that we're seeing in cells. So just thought it was an interesting analogy to that functional mechanism. So very basically, we do have to separate the inside of our body from the non-sterile outside, and that requires some pretty established lines of immune defense. Obviously, we know that we get sick fairly often at times, but we could get a lot sicker if we didn't have this innate protection mechanism in place. Innate protection is provided by what I just generally will call innate immunity, and its goal is either to prevent or at least slow down the the processes that are established by incoming microbes and any other foreign material that wants to gain access to the body. And so if you look at innate immunity, there are two major lines of defense. Okay. This here, my voice. So you may get rid of that bar for cup. I noticed there every day.
With more. I know how to do this now.
I tried to avoid titles just for that reason, but it's
I
enough to do that.
floating eating controls.
Oh, yeah, one more down.
There you go. One up. Thank you.
And you can move the Linda box.
So now,
It may not be shared yet because it looks like Linda's trying to talk. Or is that you? I think that's us.
This is called.
Okay.
it just doesn't switch slides. Oh. Might just be catching up. I think I've seen this before.
We can see it, James.
Yeah, it won't move slides.
Maybe if you exit from full screen.
You think after 2+ years with
Yeah.
and stuff on our, that speak out and
I see how my computer works.
The good part, this is all being recorded. Play this one. Okay,
Let's go for this one. How to go. Oh, cool. Yes.
even if we get rid of that again. This one?
yeah.
No. So it doesn't like that. Yeah.
Oh,
Guess we need that there.
yeah, just kidding.
That's the state.
Okay. All right, so we got these innate barriers, and these are anatomical barriers. So this is typically your skin, mucus, certain secretions, antimicrobial peptides. And the goal of this anatomical barrier is just that, to anatomically prevent the entrance of microbes into the tissues of the body. Works quite well. However, microbes do have invasive mechanisms. We can also get injury or burns or things like that. And then if that happens, you can have microbial invasion into the body. But it's a bad thing for us, but it's also generally a bad thing for the microbes, because then they encounter these sentinel layers of defense. And this is where they actually meet the innate immune system. They actually meet the cells and the molecules of innate immunity that are designed and dedicated to recognize and respond rapidly to invading microbes. And these are meant to be robust, amplified, and potent antimicrobial mechanisms to rapidly slow down, if not completely destroy, the initial invaders. I just want to make note here that these sort of amoeboid-looking things, these are white blood cells, and the focus of this talk is going to be on the regulation of their responses by receptor systems. Now, we do also know in many animals, we have an adaptive immune response. I've left that kind of shaded out because we're not going to talk about it, but this is really what imparts your lifelong immunity. This brings into play T cells, B cells, and antibodies. Very different mechanisms of activation, also of how it's actually working to increase its specificity, but we're just going to leave that alone because the focus of the work that I'm interested in is on innate immunity. And innate immunity is prevalent throughout the animal kingdom. Phagocytosis as a process is depicted here, and we're going to have a lot of this blocking out of things, but we'll just have to deal with it. But this is a cell known as a phagocyte, and what it's showcasing here is the ability to recognize, grab, and then eventually internalize and destroy foreign material. I want to just make mention here that this process, and it's not shown here, is heavily dependent on receptors. The cell needs to know that something is there that shouldn't be there, and it needs to react to it pretty quickly. And the cells that undergo phagocytosis are especially equipped with those types of receptors. That's really what makes them phagocytes. And those receptor systems, again, are part and parcel of what I'm interested in studying. As a process, though, phagocytosis is well known. It was probably initially a simple nutrient acquisition mechanism in amoeboid organisms, but we know in more complex animals, it's part of the innate immune response. It's not solely responsible for the detection and killing and destruction of microbes. It has a fundamental role in tissue repair and remodeling as well. So it sort of has two faces of this response. A simple analogy to understand the incorporation of this second layer of innate immunity is what happens when you breach the anatomical barrier, say with a splinter, which we've all had. This causes injury, but also it's a non-sterile assault to the body. So it gets through that anatomical barrier and it can introduce very quickly contaminating microbes into the tissue. Lucky for us, we have these sentinel immune cells in the tissue. We call them tissue resident cells. There's many different types. They're all white blood cells, and we refer to them as names like mast cells, neutrophils, macrophages, monocytes. Their initial response within minutes to hours is to basically figure out that there's an assault and to do something about it. There also is a holistic tissue response to this injury, and collectively the actions of the immune cell in the tissue lead to what's known as inflammation. Inflammation is a good thing if it's acute, localized, and self-resolving, because that's what imparts the ability of the upregulation of all the mechanisms to deal with the assault and to also rectify the tissue back to normal. So it usually goes through a phase where there's heightened inflammation, and that's potent antimicrobial, and then it turns over to a resolving phase, which then turns over towards more tissue repair and remodeling. But in those initial phases of inflammation, there's a lot going on. And there's more than I really want to or can describe here, but signs of inflammation around a splinter site are, of course, redness, swelling, and pain. And that's because of all the stuff that's going on. And in particular, there is a recruitment of blood immune cells into that tissue site. There's a lot of signals that are required to allow immune cells to creep over this endothelial layer and into the tissue. And in doing so, these types of cells, we generically we'll call them phagocytes. You can see one here undergoing the process of phagocytosis. There are other mechanisms at play here, but I'm going to focus on phagocytosis for the purpose of this talk. I'm sure this won't work, but we'll give it a shot anyway. So I just wanted to show you a cell basically coming into the site, finding a pathogen, or in this case E. coli, and then phagocytosing it. So there's a leading edge of this cell. It's an amoeboid movement. That leading edge is known as the lamipodia. And then there's a direct contact, and then the eventual internalization and destruction of that bacteria. So this is just a white blood cell. Very simple demonstration of it going through the process of phagocytosis. What you saw there is a process. It has a beginning, a middle, and an end. And it's a temporal process. It goes through a series of stages. Stage one is completely cut off. But what happens is there's a microbe, and it's basically touching the surface of the plasma membrane. And what it's doing is engaging receptors. Those receptors then propagate a response through intracellular signaling that leads to the sort of ordered degree of the stages of the phagocytic process. What I want to get through here is that for the membrane to dynamically remodel, to be able to both capture, tether, and then eventually engulf, you require what's known as F-actin filament formation. So these are components of the actin cytoskeleton, and cells have amoeboid movements and the ability to reach out and grab pathogens like this because of the ability to change globular actin, which is a monomer, and it's soluble. This is where it normally resides. You can't really notice it because it's soluble. But when they create these filamentous forms of actin, what it does is it provides sort of structure and sort of kinetic mobility to allow the cell to dynamically remodel the cell membranes. And in doing so, it can create these novel formations that lead to extended pseudopodia, grabbing on and internalization of microbes. This doesn't happen without directed cellular signaling. And so the signaling components that lead to this event are tuned into the recognition, instigation of the signals that deliver the responses that terminate in F-actin-mediated cellular plasma membrane dynamic changes. I'm not going to talk about the antimicrobial really too much, but the internalized microbe does get put in a compartment, and it's really bad for the microbe. Again, just to reiterate, it is a process. We do know that phagocytosis exists in this sort of spatial temporal pattern. And what we understand fundamentally and haven't really shown much of yet is the receptors that coordinate this response. There's initial sort of contact, and we call this the phagocytic synapse, and it evolves into sort of what we call a phagocytic cup. And there's an intimate but firm association between the plasma membrane and the target during this process. And there's constant signaling and information exchange that is happening to propagate the response all the way down to the end. The site that you're seeing that's lit up here is increased in intensity because they're just measuring, sort of prorating the intensity of the cytoskeletal network, and that's really looking at F-actin polymerization. But what's really important to note here is if you want to assess cellular signaling, you can look at it spatially. You can look and ask what's going on at each of these phases in the signaling process and how that is differentially affected by different receptor types. And the way you can do that as well is this isn't a microbe, this is in fact an inert bead. And you can take inert beads like this and coat them with various molecules of things that you want to test or explore. And you know that certain molecules will engage specific receptors, and it's just a way of visually assessing or at least promoting the process for study. This is a human peripheral blood neutrophil, white blood cell. It's been captured in this microcapillary, and there's a bit of suction force on it to prevent it from escaping. And here is another microcapillary that's holding on to this target bead that's coated with some ligand. And as you can see, this is sped up. It's about 320 seconds shown in a very short period of time. Again, the phagocytic process. And from that image, they went through and documented this cytoskeletal dynamics using techniques to allow them to image for those changes in the cell. But you can also localize that with looking for signaling molecules and those types of things. So at that phagocytic synapse, now we're going to walk our way into receptors, because receptors is really going to be the focus of this talk. Is again, you can see a white blood cell, it's contacting a microbe, in this case it's yeast. And right at that phagocytic synapse, that intentional contact between the host cell and the foreign material, the extracellular foreign material, you have receptors on the immune cell and ligands on the target. And the exploration of those different receptors and different targets has led to a wealth of information on what the recognition systems are and how they work to affect cellular responses. It's not the type of detail that I want to go into, but I do want to show this as the principle of what needs to occur. When you have the receptor binding to the ligand, well, that's great, but that information has to be shared with the cell. And what the receptors are oriented to do is transmit this information across the plasma membrane and into the cell. And one of the ways in which they do that, or one of the proximal ways that gets this thing started, is tyrosine-based phosphorylation. And so unligated receptors will not be phosphorylated, those that are ligated will. And that phosphorylation will precipitate intracellular signaling that leads to outcomes. And again, principles of how that works and what sets that off and what dictates that cellular language is a bit about what we study in our lab. And again, if you're just looking at it generically, the binding of receptors on the cell surface needs to be transmitted across the plasma membrane through some sort of cellular signaling. Tyrosine phosphorylation is by far not the only way. What that does, it sets in motion sort of these intracellular signaling networks or circuits that lead to outcomes. And we've already talked about one of the outcomes being the exchange of G-actin to F-actin filaments, which is the energy required to remodel the plasma membrane to engulf and grab onto targets. Should also be noted that that same sort of signaling network assists in cellular movement, it assists in cellular attachment to matrix, it assists in a lot of cell biological processes. Offshoots of this signaling network can also be transcriptional changes, And we do know that when encountering a microbe, not only do they want to set forward signals to engulf it, they also want themselves to be activated so that they can make other pro-inflammatory factors to recruit other immune cells, or even initiate intracellular signaling machinery involved in killing the microbe that was ingested. But the principle of receptor signaling is through these 3 steps. Studies of mammalian phagocytosis have allowed us to understand all of the receptor types. There's loads of them. In principle, they either bind the target directly, so they recognize something specifically on the target, or they bind to an immune molecule that is produced by the host that sticks to the target. So in the case of FCR or FC receptor-mediated phagocytosis, you have antibodies that are produced. They can stick to microbes, and then the FC receptor binds to that antibody, allowing it to take in through a process of opsonization the target. Dectin-1 was one I showed earlier. It's a direct facilitator of phagocytosis. It touches the microbe directly, and it binds a molecule known as mannan. Complement receptor 3 binds complement proteins, which are produced by the immune response when microbes infect. And it also is an indirect facilitator of phagocytosis. Regardless, they're all different receptors. They all look very different, and they don't all signal the same, but they do execute the ability to create F-actin-mediated polymerization as one of the terminal ends of their processes, which allows for the dynamic movements of the membranes for the capture and engulfment of targets. And the offshoots can be other transcriptional changes. They could be common or they could be distinct depending on the receptor systems we're looking at. And not to oversimplify too much, but the receptors usually don't work in isolation. There's usually a variety of receptors that are collaborating or even being agonistic to each other to sort of set activation thresholds for the internalization of extracellular microbes or targets. Okay, so another principle I just want to introduce is the collection of immune genes, innate immune genes that are present in the genome of animals. And in this case, we're going to talk just about humans as the comparative example to fish later on. So many of the genes, particularly those of the FC receptor family, are tightly clustered in genomic regions. And in this case, they're in chromosome 1 in humans. And each of these boxes is a different immune gene. So they're inherited, they're germline encoded, they're non-rearranging. You get them, from your ancestors. They diversify over generations, of course, but the sort of the haplotype that you get is what you've got from birth. It's the blueprint of receptors that you have to deal with. And within populations, there's some diversification, but usually innate genes are pretty conserved. And my point here is that in innate immunity, you often see this. You have to see collections of immune genes localized in the genome that are responsible for something like motivating responses like phagocytosis or other effector responses that I'll speak of momentarily. The classical FC receptors are over here. They were found, many years ago. In early 2000s, they found another receptor family known as the FC receptor-like proteins. I'm only pointing these ones out because they also control innate immune responses, and they do cross-regulate other responses in other cells. But the key here is we still don't know really what they all do, but they're clustered and they're involved in controlling the immune responses. So you can find blueprints for immune genes all clustered in chromosomal regions, and this is an example of the FCR family of genes. Again, skipping from the example to FC receptor-mediated phagocytosis, which do impart this response, there are another collection of genes on another chromosome that control another fundamental innate immune response in our body. And we have what are known as natural killer lymphocytes, which are cytotoxic cells, and they're not dealing with extracellular pathogens that exist outside the cell. They're in our body to scan for the health of our host cells to see how they're doing. And what they're looking for is sick cells that have some aberration that is noticed by these cells, but incidentally, they still use plasma membrane dynamics to touch, to form synapses, and to probe host cells. In many cases, the outcome is that you're doing good and just this retracts and it goes on and scans for another cell. But if this is a virally infected cell or a cell that's stressed or undergoing aberrant growth or neoplastic, perhaps becoming cancerous, there will be signals at that synapse, the purposeful contact between the membrane of these two cells that will lead to what's called the lethal hit. And what happens here is the cytotoxic granules of these killer cells will be signaled to be released into this target and kill it through a process of apoptosis. A very fundamental innate protective response that is also facilitated by receptors that recognize ligands and that instigate the cell to do things through things like tyrosine phosphorylation. I think this is just a really good video to showcase the dynamic membrane changes that occur in these cells. And I just want to show you that they're looking for host cells that are aberrant, and then the way they kill them is kind of cool. So you can see they're very mobile. They have the ability to sort of search tissues, and they're using, you know, F-actin, G-actin exchange to make these membrane dynamics, but they're not looking for bacteria, they're looking for host cells that aren't doing so well. So here's a target, and it forms this intentional contact known as the synapse. And the sicker this cell is, the longer this synapse will remain. And what's happening is this cell is overcoming an inhibitory threshold to dedicate its intracellular machinery, shown here in red, and deliver those cytotoxic granules into the target, ultimately killing it. So you'll see it's forming a synapse, and then these granules, these killer granules, which are now going to be introduced to you, are being spit out into that cell directly. And so the receiver of that will be destroyed through a mechanism known as apoptosis. Another fundamental immune receptor-mediated process that exists in our body. And they're just ganging up on that target there. Probing another one is here as well. So again, an intentional contact. In this case, it was against an extracellular microbe. Here, I mean, in this case, it is showing a T cell against an antigen-presenting cell, but we can just change this to natural killer cell and some sort of target like a virally infected cell or a cancerous cell. This intentional contact, again, places ligands and receptors, and it's the intracellular tyrosine phosphorylation that leads to the activation of the cytotoxic cell to deliver its final function, which would be cytotoxic killing. It's extraordinarily complicated. There's an enormous array of immune receptors and ligands that are involved. So I don't want to trivialize that, you know, this is the level of complexity. It's this and then more, because it doesn't happen in a single moment. It happens over time. And during that synapse formation where you saw the membrane of that NK cell sort of messing around with that target, there's an awful lot going on. But at least for sort of interpretive purposes, we can understand that there's, you know, many receptors involved on the immune cell and many ligands on the target. And I just want to say that we generally categorize those as inhibitory to the function or augmenting the function. And so we have lots of receptors in place to prevent these things from happening. And we have lots of receptors in place that try to make them happen. And what happens in cells is a real tight balance between inhibition and activation that occurs. And over time, during the formation of that synapse, it's probably initially slightly inhibitory. But if there's information based on the ligand binding that this target needs to be killed, there'll be an override of that inhibitory signal, leading to cellular mediated killing of the target. And so the activation thresholds are established, and these are sort of like the circuit breakers that then lead to the signaling that leads to the outcome. So it's immensely complicated. And again, the key here is lots and lots of receptors in the innate immune system controlling these responses. The genes that control that are in the leukocyte receptor complex on human chromosome 19. And each of these boxes represent immune genes. There's two major families that are represented here. So they're multi-gene receptor families that direct, control, and coordinate these types of responses. And all of this is innate immunity. We're not even talking about T and B cells. And just to give you an idea of what these encode, if you actually look at the sequences and get the interpreted structural protein, the main thing to note here is they're families. This is one family of immune genes. This is another one. And functionally, they have extracellular domains. Each of these are referred to as immunoglobulin-like domains. Basically, it's the sticky part of the receptor. It's what allows it to interact with the environment and engage its targets. That's the one event. The second event is how is that information transmitted over the plasma membrane? Well, it's through either inhibitory or inhibitory in red or stimulatory in green signaling. And that generic makeup of on-off switches in the immune system occurs throughout these innate immune genes. Again, encoded in distinct regions in our genome. The on-off nature of these switches is generically we can assess that by tyrosine phosphorylation. Now, both inhibitory and stimulatory receptors undergo a level of tyrosine phosphorylation, but what matters is where is that tyrosine relative to the sequence around it. So these tyrosines embed themselves in what are called motifs. And for instance, if a stimulatory receptor is engaged, the tyrosines that are phosphorylated are in motifs that basically generate molecular Velcro for the preference to stick to kinases. So kinases are preferably attracted to phosphorylated tyrosines in motifs that have a sequence like this. And again, that level of detail is really not important, but it's one of the molecular fundamental underpinnings of phosphatases versus kinase recruitment to receptors. An inhibitory receptor, when phosphorylated, is in a motif because it exists sort of as this molecular Velcro for these phosphatases. And when they're attracted, what they do is dephosphorylate downstream signaling to sort of dampen it or turn it off or abrogate it. And collectively, with the multitude of receptors involved and the various ligands, this creates a complex network of signals that are being produced. And it's really the balance of one over the other that dictates whether the response will be inhibited or the response will be propagated. So of course, all of our understanding of these fundamentals originally come from mammalian model systems, mouse and man, and obviously that's still being studied in depth today. But we do know that other vertebrates have innate immunity. Other vertebrates have innate immune cells, and those innate immune cells undergo the very same functions that I described to you earlier, like phagocytosis and cytotoxicity and that. And so the fundamental mechanisms exist in vertebrates, but the real gap is what are the receptor systems and signaling mechanisms involved in controlling it. And it's a huge question, but filling that gap, is what comparative immunologists essentially try to do in their own sort of specific way. Horrible slide, but just focus on the area that's highlighted with the dash and look at the positives in green. And what I wanted to note in the system that I've studied since my PhD, the teleost fish, they have all the complement of immune cells that you find in mammals. And if you look, from cartilaginous fishes down to jawless fishes and earlier, they're either not identified or not there in terms of the complement of immune cells that exist as part of the immune system. This isn't exclusively innate immune cells, but this is the complement of the standard immune cell types that you would appreciate being involved in instigating an immune response in these animals. So like was mentioned at the beginning, I studied fish macrophages during my PhD in goldfish. That was great and super interesting, but the one thing we didn't have was cell lines. Cell lines give you an opportunity to look for functional attributes of specific cells rather than a collection of cells that you get out of the fish kidney. Now, of course, we tried to use techniques to enrich the populations, but they were still from an animal's kidney. You did the experiment, you can never go back to that animal, obviously, because you're dissecting the kidney or the spleen out of the goldfish. What attracted me to do my postdoc in Mississippi is they had spent many decades developing cell lines from this animal, and they were developing the cell lines from its blood. So none of the experiments weren't terminal, and you could always go back to the animal and do things. And they just had done an amazing amount of work, and it was really eye-opening to me to realize that I didn't always have to deal with the fish if I didn't want to. I could just go to the incubator and grab a flask, and those would be macrophages. Or I could go to the incubator and grab a flask, and those would be natural killer cells. That was a huge advantage that I really appreciated and wanted to go work on more, rather than perfecting, ways in which you can enrich cells, which was done in my PhD by people before me very well, but there was limitations in that system. And so my main advisors down there were Nor Miller and the late Bill Clem, And this is a mini review from 1996, where it talks about how they're able to effectively generate immune cell lines from catfish peripheral blood. And I want to understate how much of an advantage this was, because in fish, it was very difficult, if not impossible, to get cell lines from all the different model systems. And so they somehow were able to do that, and they did it in ways that didn't require any overt attempts at doing transformation or viral infection. Just the isolation and stimulation of those cells drove them into various immortal cell lines. And they were able to acquire things like monocytes, which is something I was interested in studying. They certainly had B and T cells, but I went down there in 2003, and before I went down there, they were working on a study where they had actually identified natural killer cells. So as I've sort of walked you through this intro, I've talked about the fundamental aspects of macrophage monocyte-like function and a bit about the NK cells. And they had those cells, they were performing the functions, they looked exactly like immune cells in mammals. In fact, if you watch them perform their function, I didn't tell you it was a fish cell or a mammalian cell, other than the fact that it came from 37 degrees versus 22 degrees incubators, you would have a difficult time telling the difference. But what we didn't have and still really don't have is good markers and a really good understanding of the receptor systems that control the fundamental innate responses that are performed by these cells. And so as a comparative model system, it's ideal to have a functional cell population by which you can then ask questions. Okay, it's killing those targets, how is it doing it? And then that really piqued my attention and interest, and I went down there with the main question is, what controls the signaling responses that leads to the effector functions of catfish immune cells? And there was, I think, fugu genome, and maybe there was a partially annotated zebrafish one, or it was all coming together in the early 2000s, but not quick enough for me to take advantage of. So we did cDNA libraries where you get fragments and then you race them out and you take a look at them and compare them to the database and see what they were. Long story short, from one of the cell lines, we found 3 cDNAs, and they encoded these receptors that we called leukocyte immune type receptors. We named them that because they had extracellular domains that looked like they would stick to things. They had transmembrane segments and they had signaling potential. And if you did some phylogenetic analyses, there were some features of them that reminded us of receptors that were in mammals. So there was some nature of these receptors that were fairly consistent with those in the FC receptor complex and the LRC, so chromosome 1 and chromosome 19 in humans. And that trend existed through amphibians, birds, and into mammals. So there was a collection of information that made us more interested in these, because there were other receptors at the time that were found in fish, but they were fish specific. They had no phylogenetic relationship with any other receptor systems other than in fish. So we thought that this would be a good model to work with, because it's sort of a vertebrate model rather than just a fish specific one. Well, I thought I had 3, but there's way more than that. So this is the old school way of looking at the complexity of the family. And these are southern blots. So you take genomic DNA, you digest it up, and each of these rows is representative genomic DNA from sibling catfish, so they're brothers and sisters, so to speak. We got these from one of the hatcheries. And what you notice is there's many more than one copy, and the patterns between the individuals is diverse. So this means it's polygenic, so there's more than one copy. And there's way more than one copy, obviously, because each of these smears and bands is a different gene. And these are probes against some of those extracellular domains. And that's what each of these subplots are showing you. And if you ran this next to another polymorphic family that we're aware of, known as MHC class 1, these were as polymorphic, if not more, than what we see for that gene family. And so we realized that we weren't just dealing with 3, we were dealing with a family of multiple members, reminiscent of what happens with the clustering of chromosome 1 and 19 in humans. More recently, with the advent of genes, the genomes, we're able to go in there and mine the genomes and pull out a bunch of these forms. And what you can see is there's just lots. And most of them in catfish are on chromosome 7, but we've also found them in zebrafish, goldfish, fugu. They're pretty ubiquitous amongst teleos fishes. But wrapping your head around what to do with all of this information, what to do with all of these genes, how to study them is pretty daunting. And so when you start up a lab, you're not going to say, well, I'm going to figure out what these guys do specifically in every single form. And the key questions are, what do they bind, and then how do they influence intracellular signaling and effector responses? And so there's a lot of reagent development that needs to go into play, and we've dabbled into that, but I'm going to give you an example of how we study their signaling. But before I do that, I just wanted to remind you that the LITRs that we found, at least the ones in these green circles with each of these lines, these are LITR clusters, and they're linked to these other genes which are not LITRs. And all the LITRs in the other fish species are also linked to the same genes. And if you look in humans on chromosome 1, those linked genes are present, and they're next to the classical FCs and FCRLs. This is telling you that the LITRs, at least some of them, are synthetic to human chromosome 1 encoded innate receptors. So they're phylogenetically, structurally, maybe functionally, we don't know precisely, but they're definitely synthetically related. And that, that was an interesting thing for us to find. We thought they would be actually associated with the other gene family on the LRC, but they actually were much more close to this one. There's many more pockets of LITRs in the genome now, and they definitely we haven't found their placement just yet. This was the other gene family we thought they'd be related to. Now, what this tells us is nothing more than the synthetic relationship, but that has nothing to do with function until you try to figure that out. And so I just wanted to give the analogy that we're working on a gene family in fish that is as organized and potentially as complicated as what you see in the mammalian genome. Okay, now to how do we study them? I'm going to give you an example of my sort of pragmatic mind and also my fear of going down the path of trying to do too much with these receptors and really getting no information. So I'll just say we spent a lot of time working on the extracellular bits and have zero information about what these receptors state. Okay, fail. But I don't want to put students on projects that have fail on it. So what we did is we decided to go with a little bit more likelihood of getting some data by looking at the signaling transmission from the tails, because tyrosine-based phosphorylation and the propagation of events from there will happen. It's just a matter of us developing a system to see it and then interpret what's going on. So we use it as a model to allow students to ask directed questions about what do tyrosine-rich tails do when encoded from fish relative to what we understand what they might do if they were coming from a mammalian receptor. And so we know the organization and orientation of these tyrosine motifs. So we just developed strategies to clone them into expression constructs and put them into cell and then stimulate them and look for their function. I will say though that we have absolute confidence in that this is a classic stimulatory receptor, and we use this as our guide or positive control to make sure our systems are working. There's not too many anticipated surprises by these tandem tyrosines in an ITAM, and they've never really let us down. They've done everything you'd expect it to do. It's a classic conserved on switch of immunity. However, receptors like this and several others have unique tyrosine containing regions, unique in that they do very funny things with those tails, and we want to see how that actually helps to fine-tune signaling or instigate it in the first place. So one of the receptors we spent a lot of time on, basically to study and understand its signaling potential, it has a 6 tyrosine-rich intracellular tail. 3 of those motifs are embedded, or 3 of those tyrosines are embedded in motifs that we recognize. We have predictable suggestive evidence based on just the motif that it's likely going to recruit a phosphatase and dampen an immune response. But it has also this very unique 3 tyrosine containing rich motif that really didn't have any super recognizable motifs. What was more interesting about it is it appeared that the receptor, when we cloned it, we found a version that had or didn't have that piece. So it looked like what was happening is this was an insertion or deletion, and we wanted to know what was the consequence of adding this or removing it. And so a lot of our studies, at least of my first PhD student, was really designed around functionally characterizing the signaling potential of this tail. And the major difference for us that excited us was the fact that this piece seemed to be inserted or deleted by splicing. Again, that's odd. Most of the splicing we've seen in these receptors occurs with the removal and insertion of these domains, but we didn't really follow that up. So my first PhD student, Ben, super long story, really short. is he found that those predictable motifs did recruit phosphatases to attribute to its functional response to inhibit cellular cytotoxicity. But this tyrosine 453 unexpectedly and uniquely recruited a kinase known as CSK. What CSK does is it phosphorylates another major kinase to close it into an off conformation. And he had done an awful amount of work with natural killer cells to show the phosphatase independent and dependent functionality of that tail. And so we were excited about that. It got things moving. It showcased that we can get systems to study and gain information from the signaling potential of these receptors. So again, moving forward, we continually relied on the functionality of this receptor to help us further understand signaling potential of this unique one and others. And we called the domains that it contains the transmembrane proximal. This is the one that's inserted in and out. And this is the mainstay inhibitory part. So we've done not just cytotoxicity, we've also done some work with phagocytosis. So for the phagocytosis work, what you have to do is, because we don't know the ligands, you put a tag on the receptor. It's an epitope tag, a very short sequence. And that allows you to buy antibodies against it. And we use that to coat beads, directionally putting the Fab fragment on the outside. So when that fragment meets that epitope, that stimulates the receptor artificially, allows us to look at what are the functional consequences of activating those intracellular motifs. And what we saw predictably with the activatory receptor is that it allowed the cell now to bind and engulf beads that were coated with that antibody. So again, this is for system development more than for gaining any new information. And you always want that reliable control just to make sure that you're getting some output. If you coat the beads with an irrelevant antibody, none of this happens. It needs to be directed towards that tag. And then we can image the cells and see, it's taken up a lot of beads. We use rather large beads, 4.5 microns, so they're a bit of a challenge to be eaten, but that means the cell is really dedicated to getting that bead inside, because E. coli is like half a micron. This is quite a big, substantial entity. And I just wanted to say that we trace the process so that you can identify, if you wanted to look at signaling now, you could look at the signaling exchange at that synapse throughout the process. And we've done things like pharmacological work, and you can stain that synapse for the presence of functional molecules. So if you want to study that this is in fact a standard stimulatory pathway, you can do that. And that's what we have done. We did some SEM imaging just to show some nice attachment and internalization. This is at increasing incubation periods. And then at the end of the process, the cells are just absolutely stacked with these large beads. You want to completely arrest the process. You add a blocker known as cytoclasin D or latrunculin B, and that prevents apoptotic dynamics from occurring. And so the beads aren't even really attaching. You can trace back with more pharmacological inhibitors, and you can actually trace the pathway and see who's involved. And we've done that, but I just want to show this because it's a very visual process. And you can do some empirical tabulation of the behavior of the cells, and we just basically, I didn't anyway, but my student counted 700 cells, 930 or 401, and came up with what's called phagocytic phenotypes. And under the regular conditions, 62% of the cells are phagocytic with this receptor. The irrelevant antibody, none of them. And if you cool the cells down, these are mammalian cells, and you cool them down, the activity is diminished, and that's expected, and that's just another control. So not too much of a surprise. Horrible slide again, I apologize, but my students got tired of counting, so they wanted to automate this process. And so Marion and Harry developed this procedure to get everything onto the image stream flow cytometer. An image stream flow cytometer takes a picture and tabulates the cells in about 3 minutes. It can do 5,000 events. So as you can see, you can get high throughput automated information from this. And this is sort of the masking procedures and all of the software that they use to establish these phagocytic behaviors. And essentially what this allows one to do is to rapidly compare receptor types and mutants and different things and get data that leads towards established studies based on predicting what may or may not happen based on the composition of the receptor at play. So it was incumbent upon the students really to decide that they didn't want to count under a microscope anymore. And this adventure led to a really cool procedure that I think has been left with us from past students to allow them to study these receptor systems in a more high throughput manner. But I don't want to focus on sort of that numerical data. I want to look at something more visual, because I think it's tells an interesting story, and it also allows us to see some cool things that are happening with these cells. And I did spend a lot of time talking about F-actin dynamics, and so the reason I did that, and I should probably reiterate, we have stained these cells with a marker for F-actin, and where it's more green is where it's more intensely occurring. And most cells will have F-actin just under their plasma membrane, and that gives them their sort of their cell structure and shape. So these are both the same cells, but one is transfected with our standard receptor that we use for stimulation, and this is the weirdo one with the tyrosine-rich tail. And we notice clearly, surprisingly, and confusingly, under the microscope, that these cells are doing something strange. So clearly, just by stably expressing that one receptor type, we changed the phenotype of the cells. They were sort of producing these F-actin-rich protrusions. That was noticed, not understood, but again, we science is very observational, and from there you can kind of march forward to trying to figure out what's going on. Because all the phagocytosis I talked about earlier started with the contact already made. But we do know that the membrane dynamics of cells occur prior to that binding. So this is teasing an immune cell with a target, and it really, really, really wants it. And so you can see the nature of the cell is to try to get a hold of that target prior to it touching the surface. And if you let it touch the surface eventually, I wish I had that actual setup. Okay, so that's great. But this underlies that cells don't, especially immune cells, based on looking in the literature, they have active membranes. And those membranes are developing these F-actin-rich membrane protrusions to really increase the possibility of interacting with something that may, pass by. And what it's intended to do, hopefully, is push receptors out there that are capable of binding those targets. And then you need some sort of retractable mechanism to bring it back to the cell surface, tether it, so hold on to it, and then probably pass it over to another receptor to engage the engulfment process. This has a name in the literature called tethering and tickling. It's actually in titles of papers, but this would be the tethering. And then the tickling is when you stimulate the receptors to undergo the engulfment process. But that observation that we got from those two constructs led us to this sort of appreciation, and we wanted to try to figure it out a little bit. So, if you have an external target and it's just bouncing around through Brownian motion, sure, it may hit the membrane. If you have a flagella on a microbe and it's swimming, it may bump into the into the immune cell. But if you protrude your membranes, you increase the chances, and you certainly increase your surface area of making contact with extracellular targets. One of my students gave a 3MT on this, and he equated it to if an octopus is just sitting on the bottom of the ocean hoping a crab will crawl over it, it will starve. But if it actively protrudes out its tentacles and its arms, it will have a higher chance of grabbing those targets. Wish I would have thought of that. And you can see from SEM images close-ups that there are these tethers that do form to touch the microbes. And here's another tether. And this one at the end shows you the remnants of the tether after this tether had retracted back to the cell. So it sort of lassoed it and pulled it in. And another way of seeing that is through these images here. This is a target. These protrusions have touched and then retracted. So we wanted to see were the protrusions developed on those cells just randomly flailings of the cell, being engineered by our receptor, but did they have functionality? Were they decorated with the receptors that allowed them to touch and pull those targets back to the cell surface? And again, this is reminding us that this had this awkward, weird phenotype that I've just been talking about. And so when we place the target beads next to the cell, I've kind of spoiled the story down here. What you'll see is you'll notice there are times where those beads are just sitting there, And I don't know if it's incidental or on purpose, it's probably incidental. It makes contact and then retracts. And then it seems to go a secondary wave that tethers the target to the cell surface, but it doesn't engulf it. So it's actively captured and tethered. Okay, this one is another example, but this is more of a skinny projection, touches the target, brings it to the cell surface. Now remember, those beads are coated with anti-HA antibody, and those receptors are decorated with the epitope tag for that antibody. So this is a specific response. If you use the IgG3 beads, the tentacles aren't interested. They may brush up against the bead, but they don't retract it in. So we are fixing these protrusions to make contact with these target cells, these target beads. And again, if you just look at this one over time, you can see the protrusion comes out, makes contact, sticks to, retracts, and then you see this secondary wave of actin. Doesn't cause the engulfment, but the bead sits in this firmly attached position. It is formally tethered to that cell. Now we haven't equipped these cells with a counterpart receptor for the tickling action, which may actually have brought it in. But we never intended this to be a point of study in the first place. Again, tethered, retracted, and then fixed to the cell surface. And when we took ultra SEM images at our microscope facility downstairs, we could see absolutely you're getting these formed tethers that are making these structural sticking points to the target. What I liked about this is here's an actual phagocytic cup. And the reason it's available is because it detached during the processing of the cells, but this was also stuck to that bead. And so in these phenotypes, we saw a lot of sharing of beads between the different cells. But for us, the noticing was these thin tethers, these thick tethers. This seemed to be a matured anchorage point where it was firmly fixed to the cell surface. And again, throughout this, you can see these phagocytic cups where the bead used to be placed, but I think there's some dehydration that occurs here. And over time, from left to right, you see the early formation of the tethers. If you look real close, you can see the structural contacts and membranes stuck to the surface. And then eventually it terminates in these retracted phagocytic cups, and it just sits there. Doesn't go any further, just sits there. We leave it for an hour, they're still sitting there. Some of them do slide in, but very, very few. And so we believe that what we're seeing here with our receptor is this process: the probing, the attachment, the retraction, and then the tethering. So this is much earlier than we anticipated. We were always thinking we were going to be studying just engulfment, but this receptor provided us an opportunity to study this process. Okay. I just want to say now what our goal was at this point was to do more studies to try to come up with an idea about what's making this work. And I'm not going to show the data on that because I'm way over time, but I will say that we had 2 candidate molecules, which were through a lot of data, looking through the literature and whatnot, and then formulating actual experiments to test it. We had already figured out that this binds CSK, but this adapter here is known to directly link immune receptors to the cytoskeletal machinery. And we thought that this was the process that allowed the constitutive formation of these protrusions. And then what we decided to test is whether or not after engaging the target, does the receptor then further undergo recruitment of this enzyme SYK to sort of firm up that tethering. And a long story short is that's where we sort of ended up. And the model systems that we used to study that was a lot of immunofluorescence and immunoprecipitations and those types of things. And forget all this is just staining for those targets. We were able to actually co-locate in those protrusion positions both the presence of SYK and the presence of NCK, but most notably, only NCK was there when the targets weren't present. So the constitutive deployment of those protrusions required that first recruited molecule, NCK, and then the tethering required the secondary recruitment of SYK. The SYK recruitment to this receptor is very unusual because it's actually recruiting it to inhibitory motifs, but they created the spacing that allowed this to occur. And we were very excited when we saw SYK recruitment at this receptor in the position where the beads were bound. So after it made contact with the beads. And I apologize for timing. And just for some general, I guess, like application, like there's no formal application for this. This is the study model. This allows the students to think about, research questions and try to interpret what's happening with their results. But cells make protrusions all the time, not just immune cells. And in particular, metastatic cancers are known to make what's called invatopodia. And they make these F-actin-rich protrusions, but they decorate the surface of those with enzymes. And this allows them to embed themselves in the extracellular matrix and actually invade into tissues. So the studies about understanding what deploys them and what function they have extends just beyond simple LITR model system. We've maybe contributed the idea that some of the components of that are likely NCK and SIK, and that can happen in non-traditional ways. And again, this is reiterating the F-actin mediated deployment of these enzyme-rich filopodia into tissues. Ours was F-actin-rich deployment of cellular structures to bind, grab, and tether targets to the cell surface. And F-actin polymerization is fundamental in a variety of cell processes, and I just wanted to acknowledge that phagocytosis is not only a good model system for studying immunity, it's also an excellent cell biology model system for studying aspects of signaling that leads to anything to do with F-actin polymerization. And we've touched on cellular movement with the lamipodia, we've touched on the filopodia, the invatopodia, phagocytosis, but there are other events as well that occur mediated through F-actin polymerization. And then just final slide, all the work that I showed today has been done by students that have worked on the catfish LITR system. It's been a real pleasure to have such talented and dedicated students in the lab. We've moved on now, fortunately, or fortunately, I don't know, from catfish system. And we're now working in both zebrafish and goldfish to think about new ways and new strategies to try to understand what these receptors are. And we have zero information on their importance in the actual animal, but that's not the direction that I chose to go. Thanks for your attention. Sorry for going over time.
Great, thanks so much, James. So if the floor is open for questions, and then I'll just open this up here to the chat section because there may be some questions there as well.
James, I wonder if you'd look to see if these receptors are involved in endocytosis of like a smaller particle than.
Yeah, the smallest speed we've used is 1 micron, and that's not endocytosis. We do know the receptors are endocytosed in recycling. So you cross link them on the cell surface, they'll go in. So we know that happens, but are they actively being used to bring something into the cell? We just don't have, we haven't addressed that specific, but they can endocytose themselves.
Okay.
Just, yes, normal turnover, right? Sure.
And then there's one from the chat.
So when defensive cell phagocytoses a bacterium, does it manage to get any nutrients out of that bacterium, or is its job just to destroy the thing?
Yeah, so for me, when I think about that, that's a good question. I don't know what happens to every single molecule that is generated through the destruction. I imagine some of them could be picked up, absolutely. What I do know is what they're not just expelled from the cell, many of them are packaged onto receptors and reintroduced back on the surface to present that information to the helper T cells. And so antigen presentation is a major outcome of the degradation of those microbes. Are there bystander or even active ways in which the cell can recoup some of those sort of so-called nutrients? I bet there is. But off the top of my head, I don't want to guess because I don't have a piece of literature in mind to support that.
Thank you.
So Ted's question, so great talk, James. Have you tried any ancestral reconstruction to try and parse out the history of the homologs showed by symptom?
You mean go behind fish, like in like cartilaginous fish and skates and things like that? Like if you're asking, like if we look prior to fish, it really, in my experience, it really deconstructs. You can see egg domains in receptors that have, I think you have to squint really hard to say that they're related to the LITRs, but it really, for me, holds the most consistency at the level of the teleost. But once you go into sharks and skates and then anywhere beyond then there, you know, when we were looking back in those studies, we didn't see anything that, you know, there are receptor systems in those animals and they have big domains and they have the signaling motifs, but making them LATR relatives was not easy.
Okay, thanks.
Yeah.
Great
You
talk.
know, it's very interesting that you have the one receptor that only mediates up to the point of tethering and maybe just holding on.
Yeah.
And then you said that, you know, then you need tickling in order to engage other components in order for the endocytosis or phagocytosis to occur.
Yeah.
Now, is there a biological relevance to a cell if the process just stops at the tethering and the lassoing and holding on part? So I'm just wondering that you know, it doesn't necessarily have to, everything has to go all the way to engulfment and internalization.
So the way I would, the analogy that I always thought of with that is we're using phagocytosis as a surrogate model to understand what's going on. So interpreting the outcome as that receptor is meant to tether stuff on its surface. Well, we forced it to do that because of the model that we're looking at beat capture. But maybe this is something I didn't really talk too much about, is the maturation of immunological synapses is a graded process that requires a multitude of receptors. And a lot of those are tied in to forming a central structure that is dictating the signaling, but also forming a sort of a side structure that's firming up that connection, allowing the cells to stay together longer and for the necessary time for the adequate signals to be, relayed. Particularly if you're forming a killer synapse, you don't want those cytotoxic granules to have any chance to, get out and be randomly destroying bystander cells. You want to direct them into the target. So with that tethering, I always thought that maybe that is showing us that type of receptor, and if it's only in isolation, is using actin membrane dynamics to affect the contact time that a cell might have with another cell. But because we only used a bead, we just saw it tether and stick. But maybe that's part of an internal process during synapse formation to allow the maturation of the actual synapse itself. And the reason we make the conclusion that it's an attraction and a tethering is because during synapse formation, you remember that NK cell was interdigitating and very dynamically remodeling on the target. I think what it's doing is it's finding a place to sort of share that information and firm up that contact. And we do know immunological synapses that are stimulatory stay with the target cell a lot longer than the inhibitory ones, which disengage rather early. So maybe that's a proxy for how actin dynamics affect cell contact with something other than a bead, perhaps with a target cell, perhaps with another immune cell. And not to forget that we do know that these receptors cooperate with each other and antagonize each other. And so I do think you need the appropriate complement of receptors to truly understand what they do, which is why I think it's been very difficult for us to find the ligands, because we don't know which receptor combinations we can choose to create the platform for how they interact with their targets. Long way of saying I sort of really don't know.
I'm just sort of thinking that maybe this tethering is also, as you said, to enable contact to be maintained for more extended period of time.
Yeah.
So that other information can be passed between the two cells.
Totally. Yeah.
Not just sort of in terms of phagocytosis, but other signaling intercellular signaling.
Yeah.
That's a normal component.
And historically, that receptor was actually cloned from a cytotoxic cell line. And that piece that goes in and out occurred when that cytotoxic cell line was stimulated to the target. So by analogy, you can think that our phagocytic system just showcased its ability to affect F-actin dynamics, but it's not even interested in beads or extracellular targets at all. That's why I think the analogy at the beginning was to showcase that membranes can be used to form contacts or find extracellular targets.
So Andy, and then we'll probably stop there.
So is there anything that's homologous in invertebrates? I mean, they have phagocytic cells. And they'll engulf bees, and they'll send out those long extensions all the way across the country.
Right.
So is it a similar mechanism?
Well, I mean, we do know that NK cells in mammals create tunneling nanotubes, which we studied them in flasks to allow the cells to sort of literally be able to bump into each other in solution. But tissues don't allow
I'm just
that.
saying in invertebrates, if you keep a group of phagocytic cells, an overwhelming amount of bacteria,
Right.
they don't react to individual bacteria as phagocytic, they don't phagocytize the bacteria. What they do is they form a clump and then they fire out all their DNA and make a DNA net around all the.
Yeah.
Around all the, so is there a parallel with the system?
I mean, fish cells make nets, that's well understood, and I mean, so the way I'll end this question is, the one reagent that we have made in goldfish is an antibody against one of the LITF forms that is stimulatory. And we use this antibody against fish cells. And that antibody actually lights up fish neutrophils, goldfish neutrophils, only on Western, of course. We can block it with the peptide, but what would help answer your question is if we could get that antibody to work on the neutrophils and cross-link it to see if it allows for the deployment of nets. I mean, we've looked at cytokine secretion, degranulation, and phagocytosis as model systems to understand the signaling because it gives you an effector readout, but none of this is meant to predict what they actually do in the actual animal because none of that's been tested by me. The Mississippi group has done more with that, but no. I don't have a direct parallel to that.
Well, with that, I'd like to thank you very much for a wonderful talk today, James.
Thank you.